Today, we're diving into something that feels like it's straight out of science fiction. We're talking about a new tool that's learning to read the language of life itself, our DNA. It's called AlphaGenome, and it's teaching us how to read the book of life.
Just let this sink in for a second. We've mapped the entire human genome, right? But more than 98% of it is still a total mystery. It's this genetic dark matter. We know it's not making proteins, but it's packed with all these switches and dials that control everything. So what if we could finally figure out what all that so-called junk DNA is actually trying to tell us?
So here's our game plan. We're going to start with our DNA's biggest mystery, talk about a huge trade-off that's been holding research back, and then introduce AlphaGenome. We'll see how it's totally raising the bar, watch it solve a real genetic puzzle, and then we'll look at what's coming next.
Okay, first up, our DNA's biggest mystery. Let's talk about this genetic dark matter and why figuring out this non-coding DNA is like one of the biggest challenges in all of biology right now.
This is the number to remember: over 98%. That's how much of the genetic variation between any two people is found in these mysterious, non-coding parts of our DNA. So if we ever want to truly understand the genetic roots of most diseases, we have to figure this stuff out. It's that simple.
So why is it so ridiculously hard? Well, for one, it's not a simple recipe for making a protein. It's more like a massive, unbelievably complex switchboard that's telling our genes when to turn on, when to turn off, and how loud to be. It can physically change how our DNA is folded up, and its effects can be super specific, like only happening in one tiny type of cell in your brain. It's a whole symphony of regulation, and for decades we've basically been trying to make sense of the sheet music.
And to try and crack this code, scientists have been stuck with a fundamental trade-off. You can think of it as a choice between seeing the forest or seeing the trees. This one dilemma was really holding back progress.
Okay, here's how the problem worked. On the one hand, you had models that could see the forest. They could zoom out and look at these huge stretches of DNA. We're talking 500,000 letters, to see how different parts might talk to each other from far away, but the picture was really blurry. On the other hand, you had models that could see the trees. They could zoom all the way in with perfect clarity down to a single DNA letter. But their field of view was tiny, maybe 10,000 letters tops. So they were missing all those crucial long-distance connections. You could get the big picture or the fine details, but you couldn't get both.
And this is where the breakthrough happens. This is AlphaGenome. It's a brand new, unified model that's designed to finally see both the forest and the trees at the same time.
So how on earth does it do that? Well, it comes down to three key innovations. First, massive context. It looks at 1 million DNA base pairs all at once. That's huge. It's enough to capture 99% of all known long-range interactions. Second, pinpoint precision. Even while it's looking at that giant stretch of DNA, it can make predictions down to a single letter. And third, a unified view. It's not just looking for one thing. It's predicting 11 different kinds of genomic activity all at once. It's the whole shebang.
And this is a peek under the hood at the architecture that makes it all possible. It basically combines two different types of AI. One that's amazing at seeing local patterns, and another that's a genius at spotting long-range connections. Putting them together lets it process these enormous DNA sequences and spit out thousands of super precise predictions all in one go.
Okay, so the tech sounds impressive for sure, but how does it actually perform in the real world? Let's take a look at the new state-of-the-art and see what happened when they put AlphaGenome up against the best models out there.
And the results are just staggering. They ran 26 head-to-head tests to see how well it could predict the effects of genetic changes. AlphaGenome either matched or beat the best competing models in 25 of them. 25 out of 26. That's not just an improvement, that's a commanding lead.
Let's just zoom in on one critical test, predicting if a tiny genetic change will ramp up a gene's activity or turn it down. Compared to the previous best model, called Borzoi, AlphaGenome showed a 25.5% improvement. In this field, that is a massive leap forward.
But, numbers on a chart are one thing. Let's see this thing in action. Let's watch it solve a real genetic puzzle and connect the dots from the genetic code all the way to the real-world consequence.
So we're going to look at a very specific case, a tiny mutation that scientists knew was causing the type of blood cancer called T-cell acute lymphoblastic leukemia, or TALL.
Okay, so here's the mystery that scientists were trying to solve. There was this tiny mutation way far away from a gene called TAL1. but somehow it was causing this specific kind of leukemia. And tau-1 is what's known as an oncogene, a gene that, if it gets turned up too high, can trigger cancer.
The billion-dollar question was, how? How could this one little change, in the middle of nowhere, be flipping the master switch for cancer so far away?
And this is where that unified view, that ability to see everything at once, becomes a superpower. AlphaGenome connected all the dots. First, it predicted that the mutation created a brand new genetic on switch, an enhancer. Second, it predicted that this new on switch would act like a magnet for a specific protein called MYB. And third, it predicted that when that protein landed on that switch, it would cause the TAL1 cancer gene's activity to just skyrocket. It laid out the entire biological mechanism, step by step, and it perfectly matched what biologists had already spent years discovering in the lab.
And this quote from the paper just nails it. A core strength is its efficient multimodal variant effect prediction. In plain English, what that really means is its ability to see the whole puzzle at once. All the different pieces, all at the same time, is its secret sauce. That's what lets it solve these incredibly complex mysteries.
So where do we go from here? This is an incredible step forward, but what does the horizon look like? What comes next, now that we can really start to read our genome? The potential applications are just wild. We're talking about running experiments entirely on a computer, in silico, which could speed up research like crazy. This could lead to way better diagnoses for rare diseases caused by these sneaky non-coding variants. We could even use it to help design totally new kinds of drugs, or even guide AI to generate useful DNA sequences from scratch.
But of course, the job's not finished. There are still some huge mountains to climb. We need to get better at modeling things that are even further away in the DNA. We need to capture all the tiny differences across every single cell type. We need to expand this beyond just humans and mice. And, the ultimate goal, connecting a single letter change in DNA all the way to its final impact on a person's health.
Which really brings us to the final, massive question. For the very first time, we are truly starting to read the genome's regulatory code, the operating system of life itself. So the question, for all of us, is what should we do with that knowledge? The ability to understand, and maybe one day even write, in the language of our own DNA is going to present humanity with some of the most profound opportunities and challenges we've ever faced.